All right, let's start. Welcome everyone in the webinar. Today we will talk about milk and immunity. My name is Marlotte Vonk and I'm working in the Global Nutrition Development Team of FrieslandCampina in the R&D Center, which is located in Singapore. So for today's webinar, we have two parts that will be covered. The first half will be covered by me, and the second half will be covered by my colleague, Professor Joost van Neerven. And in the first part, I will talk about the immune system to explain a bit more about the background. And in the second part of my talk, I will go into the milk components in bovine milk that influence immunity. And in part two, Joost will explain you about epidemiological research about bovine milk and also shows insights from experimental studies. After both our talks, we will cover a few questions that we received via the FrieslandCampina Institute email address. And hopefully, you'll find this interesting as well. So first, the introduction to the immune system. I guess that these days you hear a lot about immunity and several communications referring to the immune system. But to have a good understanding about this, I would like to go through a few terms that are commonly used. So from a historical perspective, immunity basically means the protection against pathogens. And when we refer to all components like the cells, the tissues, the communicating molecules, they all together is what we call the immune system. And when we look at what they do, so the coordinated response of all these individual components is what we refer to as the immune response. So good immunity is all about balance. If you have a healthy body, that means that your immune system is at balance. And there are many factors coming from your external environment, but also from your body itself that can influence your state of balance. For example, the genetic makeup, but also dietary components or your microbiota composition, but even external threats like pollution or smoking can all influence immunity in some way or another. And if you have an immune response that is inadequate, so it's basically underperforming, you can encounter diseases like infections or immunodeficiencies disorders. But on the other side of the spectrum, if you have an overreactive immune response, you can encounter problems like allergies or chronic inflammation. Even autoimmunity, like for example, rheumatoid arthritis, is a sign of an excessive immune response that makes you experience adverse symptoms. So the immune system basically has two key mechanisms. First one, innate immunity, is recognized by a very fast response upon encounter of a pathogen. So if an epithelial lining in, for example, the gastrointestinal tract or in the lung tissue, if it encounters a microbe from outside, it can pass through the barrier where it will find many different cell types that are able to take up these microbes and present parts of the microbe to cells that are lower in the cascade. And this is usually a response developing in the first few hours upon an infection. In addition, the adaptive immune system is recognized by a way more slow response time. So this can take several hours or days and even weeks to develop to its full potential. And the adaptive immune system is recognized by its ability to form memory against the microbe that was encountered. And when there is a second exposure to the same pathogen, this will trigger a way more fast response compared to the first exposure. So there are a lot of different cell types in both the innate and adaptive immune response. And just to give this a bit more clarity, I made a schematic representation of an immune response on the right side of the slide. So at number 1, you can see the encounter with a specific pathogen. This can be a virus or a bacterium, for example. Once these are taken up by antigen-presenting cells, like the dendritic cell in this case, pieces of this pathogen can be presented to T helper cells. And the gray dashed line indicates the step from the innate system towards the adaptive immune system. And under the influence of specific cytokines, messenger molecules, basically, the T cell starts to differentiate into a different subset. And these are recognized by the receptors that are expressed on their surface. So both of these T cell subsets have killing capacity themselves, but another way in which the immune response can occur is that the CD4 positive T cell stimulates B cells that start producing specific antibodies. And these specific antibodies are able to recognize this pathogen and opsonize it, meaning it attaches to the surface. And once the big cleaners of the immune system, the macrophages, encounter such a component, they know that they should clean it and remove it from the body. So when you are born, your immune system is not fully up and running yet. The innate system is much earlier at its potential compared to the adaptive immune system that needs even several years to grow to its full capacity. But in addition to these two key mechanisms, there's also a way a newborn is protected via passive immunity. So what does passive immunity mean? Well, the infant, when it's still in the womb, it already receives protective antibodies via the mother. So the mother's immune system creates IgG antibodies that are able to cross the placental barrier and go into the circulation of the infant. So upon birth, the infant has these already in its own blood circulation. And of course, the levels go down after birth because the mom doesn't supply anymore in these IgGs. But because of breast milk, components like IgA increase in concentration, and also the infant's immune system slowly starts to produce its own protective components. However, this, as mentioned, this will take a few years to become fully functional and strong. So not only early life is recognized by a bit less immune resilience, this is also, for example, the case in times of pregnancy and when an individual is aging. So over the course of adult life, the immune response reduces a bit in strength. And this can, for example, be caused by a lower number of specific B cells and T cells, or a lower specificity of these protective antibodies. In any sense, all these effects will add up to a decrease in the strength of the immune response. And of course, aging is not something you can easily prevent. We would all like that, of course, but there are things that you can do to keep your immune system as strong as possible and as long as possible by undertaking physical exercise and also making sure you have a healthy diet. Both of these factors have been shown to positively affect the aging immune system. So having looked at aging as an influencing factor of immune resilience, dietary components can actually also influence immunity. And a lot of these lessons we have learned from looking at the composition of human milk. In addition to providing nutrients to the infant to make sure growth and development is optimal, the human milk contains many immunoactive components. And the transfer of immunity has already been recognized in the very early days when scientists discovered that if you vaccinate mice, the subsequent breastfeeding is the reason that there's also protection to the pathogen in the pups. And when you look at the composition of human milk, the protein fraction is a rich source of all sorts of bioactive components. And in the second part of the webinar, Professor Joost van Neerven will go into more detail into bioactive components in milk. So for now, I will leave it there, and I would like to zoom into another dietary component in human milk, the human milk oligosaccharides that have immune effects upon ingestion by the infant. So the HMOs, the human milk oligosaccharides, are the third largest component present in human milk, And these are carbohydrate structures that are not being digested by the infant's own gastrointestinal system. So they reach the gut in a more or less intact state, where they are fermented by the microbiota that are present, and thereby they produce all sorts of metabolites that also have an effect on the immune system. But overall, there are two key features that are recognized for HMO function. And the first one is that the pool of HMOs together are responsible to shape the gut microbiota composition in newborns, and thereby have an effect on gut health and also the development of the immune system. In addition to that, in vitro studies have so far shown that HMOs can act as a decoy receptor for pathogens in the gut. And this gives the hypothesis that this is how they reduce the risk of infection in the gut. So going one step into this prebiotic effect that I just mentioned, looking at the most abundant HMO in the total pool, 2FL, this one has been shown in vitro to stimulate the growth of bifidobacteria. Bifidobacteria are recognized by being the strains that are the most present in the infant gut in breastfed infants. And in this experiment, it was shown in the left side of the figure that the growth of 2 bifidobacteria strains was higher compared to the growth of 2 types of pathogens. And in the other side of the figure, you can see that the consumption of 2FL was much higher by the beneficial bifidobacteria strains and was almost absent in the 2 types of pathogens that were tested. So the second key role that I just mentioned, the effect on infection, has been shown in, for example, this association study. So the total pool of 2 linked fucosylated oligosaccharides, which includes also 2FL, they were measured in human milk samples in Mexican mothers, and they were the association was investigated with the amount of diarrhea in the infants that were enrolled in the study. And what they showed is that the concentration of these two linked fucosylated oligosaccharides were associated, the higher their concentration, with the risk of diarrhea in these infants. And on the right side, you can see, just for clarification, this decoy receptor effect that I just mentioned. So the idea is that the gastrointestinal epithelial lining, this expresses certain glycan structures that are being used by the bacteria to enter the gastrointestinal tract. And if there's presence of these HMOs, like for example 2FL, these can already bind to the pathogens, making sure that this interaction with the glycan structure cannot occur, and thereby the pathogen doesn't have its entry into the tissue. So in summary, for this first part, the immune system is the responsible system for the protection against harmful threats from the outside environment. And in order to maintain healthy, the immune system should always stay as much as possible in balance. And in early life, as well as in older age, immune resilience is a bit less optimal compared to a healthy adult. So that brings me to the second part of the first half of the webinar, in which we are going to have a look on bovine milk components and how those influence immunity. So first of all, it's key to have a well-functioning immune system to make sure that you have the optimal protection against infections. And what can you do yourself to make sure your immune system has everything it needs to perform its regular tasks? Well, for example, having a healthy diet, but also having physical exercise and making sure you have enough sleep, all contribute to your overall health and thereby also to the function of the immune system. And of course, healthy diet probably sounds familiar, but contains of vegetables, fruits, some animal protein, and also, for example, dairy products. So overall, the consumption of milk is recommended as part of a healthy diet. So looking at milk, in this case, 1 glass of milk of about 200 ml, we can see that this is a very nutrient-dense liquid because 1 serving of milk gives you the amount of protein that is found in 1 egg. It has all these vitamins and minerals present, like vitamin B2, vitamin B12, just in 1 serving. And also, it's a rich source of, for example, calcium, phosphorus, iodine, and potassium. So looking at bovine milk, we often highlight the protein in bovine milk being of high quality. High quality protein is needed to deliver the building blocks for all your tissues and cells in the body. So not just to build your muscles, but also for bone function. And in addition to that, the body is always continuously renewing all your tissues and cells. So there's basically a need for a continuous supply of amino acids as building blocks to make sure that all these processes can continue. And the immune system in that sense is no difference because basically all immune mediators and cells are made of protein. So in that sense, the rich supply of amino acids is key as well. And from research, we know that if there is for a longer period of time, a too low protein intake, that this can lead to a decreased immunity. So individuals with a low protein content of their diet can be more susceptible to infections, for example. So how do you determine whether protein is of high quality? Well, there are 2 factors that you need to take into consideration. And the first one is the concentration or the amount of the essential amino acids. And the essential amino acids are the ones that your body is not able to produce itself. So they should be present in the diet to make sure the supply is sufficient. And in addition to that, the digestibility of protein, so how accessible are these amino acids to your system, is also important to determine quality. And there's an internationally recognized scoring system to rate protein quality in different food sources. And both the whey proteins and the caseins, which are the major bovine milk proteins, score very high in this grading system. So in addition to protein, the immune system relies on vitamins and minerals to perform its duties. And if you have deficiencies in your vitamin or mineral intake, this can be associated with immune disorders in the end. And if we look at, for example, calcium, which is very rich in milk products, calcium is basically needed at the very, very basic level of cell function. Because a cell, once it is stimulated via its receptor on the outside, there is a very fast influx of calcium into the cell to activate all sorts of downstream processes. So calcium is basically at the start of every immune response. And in addition, both vitamin D and vitamin A are being described very often in relation to immunity. And those are playing a role in many different processes, like, for example, epithelial barrier integrity, but also tolerance induction to food proteins, and also, for example, the production of antibacterial peptides. Well, by nature, bovine milk is a good source of vitamins and minerals, as I just showed in the slide with all the food products. And in addition, for example, vitamin A and vitamin D are also present in bovine milk, but to further increase the content, these are often supplemented in commercial milk. So the take-home message of part 1 of the webinar is that an optimally functioning immune system is the key to protect against infections. And to help your immune system do its job, basically, milk protein, but also vitamins and minerals are important elements to make sure the normal function is taken care of. And in addition, there will be a more emphasis on milk bioactive proteins with their specific immune effects in the second part of the webinar. And before I close this part, I'm gonna go into a few questions that we received via the FrieslandCampina Institute email address. And one of the questions that was addressed by one of our participants is cow's milk allergy the same as lactose intolerance? Well, the short answer is no. Cow's milk allergy and lactose intolerance are distinct physiological reactions. When an individual suffers from a cow's milk allergy, there is a continuous immune response upon ingestion of cow's milk protein. And if an individual suffers from lactose intolerance, they don't make enough of the digestive enzyme lactase that is needed to digest lactose, the most important milk sugar. Well, cow's milk allergy usually occurs in the first year of life, and in the majority of the infants, this is outgrown naturally. Of course, not in everyone, because a more resistant phenotype is also possible. And lactose intolerance is something that can last a lifetime. So the second question that we picked out was whether goat milk is as helpful for the immune system as bovine milk. Well, there's no clear yes or no answer. It's a bit of both. The total protein and mineral content of goat milk and bovine milk is more or less similar. However, we have no insights to date whether the specific differences in the bioactive proteins that are present in both milk types or the subtle differences in the microcomponents, whether they affect health outcomes or not. What we do know is that goat milk consumption was shown to be safe in both infants and in adults. So for that, there's no concern. But there is definitely a need for more conclusive evidence to confirm whether there's indeed a difference in digestibility and also whether there's a difference in mineral absorption between the two. And the last question I would like to cover was, is a plant-based substitute as helpful for the immune system as bovine milk? Well, again, no straightforward yes or no answer. But if you compare, for example, bovine milk with a soy drink, then the total amount of protein is more or less comparable. And soy drinks often have the vitamins and minerals supplemented to it. But here you should always keep in mind that also the food matrix is a determinant factor for the bioavailability of micronutrients. So it's not always simply to supplement. If you compare bovine milk to, for example, oat drink or soy, oat milk or almond drink, then there is definitely an advantage of consuming bovine milk. So with that, I would like to close the first half of the webinar. Thank you for your attention so far. Please stay tuned, and then I will now hand over the virtual floor to my colleague, Professor Joost van Neerven. Thank you very much.
Well, thank you, Marlotte, for your very clear presentation. I would first like to introduce myself. My name is Joost van Neerven. I'm an immunologist. I work at FrieslandCampina R&D Center in Wageningen, as well as Wageningen University. What I would like to do with you is take you through some of the epidemiological data there are on milk consumption, and then move into the protein components of milk and their immune functionality. As you may know, and may also not know, of course, is that there have been a lot of epidemiological studies looking at environmental signals of asthma. And what these studies found out was that consumption of unprocessed milk from cows, as well as the environment on a farm, can help to prevent asthma in small children. And I would like to point out that, and it should have been below my slide, and somehow it dropped off. is that we cannot recommend consumption of unprocessed milk because, of course, what you have is the possibility that raw milk contains pathogens and induce diarrhea. But in epidemiological studies, it is possible to study children that habitually consume milk from a farm. Especially when they live on the farm, they often drink this unprocessed milk. And when the group of Eric Vermoetjes did that in the past, this is one of the first studies they did on this, and they looked at the effect of, on the one hand, no exposure to a farming environment, so not exposed to stable, but also no consumption of unprocessed milk from the farm. That's the purple bar. And then they looked at children that either had exposure to the stable environment or consumed the unprocessed milk from the farm, or were exposed to both these conditions. And what they found was that there was a clear association between the farming environment itself, but also the consumption of the unprocessed raw milk on the prevalence of hay fever and asthma symptoms. And as you can see, especially for the asthma in the bottom graph, children that grow up on a farm and also drink raw milk from the farm really have a huge decrease in asthma prevalence. Now, this was not a single study. Multiple studies have been performed on this. And I would like to point out this one study in which they actually looked at how do these children consume the raw milk. Because if you live on a farm, you consume raw milk. You can also, of course, boil it, and then you still have heated milk. But what you can see in this graph is that children that consumed the raw farm milk, but only got it when it was heated, had an odds ratio of 0.97, which means that the relative risk that they have to develop asthma is almost 1, compared to the children in the study that were exposed to drinking high heat treated milk from the shop. When they consumed the raw milk without heating, the odds ratio for asthma dropped to about 0.5 and a little bit. And that basically means that they have a twofold reduction in the risk to get asthma. And when you move to the bottom of the slide, when you take all this milk and look at the level of these proteins, and for this matter, it was looked at IgG content, so the immunoglobulin content in the milk, that the odds ratio when there was less than the detection level of the measurement assay for the IgG present in the milk, which is of course a way of looking at the heating status of the milk that they had consumed, which then would include both the heated farm milk but also the high heat shop milk children. Then, and you set that relative risk at 1, then you see that the higher the level of these intact proteins you pick up, the lower the odds ratio gets, so the lower the risk to develop asthma gets. In yet another study, it was studied if the consumption of unprocessed milk, because it has an effect on asthma, and asthma is an immunological disease in the airways. You can also assume that if you have an effect on the prevalence of asthma, you might also have an effect on respiratory tract infections. And that was studied in a follow-up study. And again, what you can see here in the different graphs, if we start at the top left, rhinitis, basically in this study is a runny nose. In children, and I should maybe address that a little bit more closely, it was a prospective cohort study in which children from just after birth, from the 2nd month, were studied for an entire year, so until the end of the 1st year of life. And at every time point, questionnaires were filled, samples were taken, and looking back at the end of the 1st year, all these data points were collected, and then the analysis was done. And when you look at the effect on rhinitis, you see that, again, when you take the children that drink UHT milk, which is high heat treated milk, as a reference, that when they, instead of UHT milk, consume pasteurized milk, you see the odds ratio for rhinitis going down a little bit. But when they consume farm milk, especially unprocessed farm milk, the risk for rhinitis, runny nose, decrease. You could see the same thing for respiratory tract infection, which is very similar to the runny nose effect in these young children. But for otitis media, so the middle ear infection, you could even see a more pronounced effect of the consumption of raw milk versus the other milk type in the study. So this indicates that not only does the consumption of raw milk associate with lower prevalence of asthma, but also of respiratory tract infections, especially upper respiratory tract infections. Now, more recently, a meta-analysis was done on the epidemiological studies that have been performed in the past. And this is just one graph from the entire meta-analysis, and this shows you the effect of current raw milk consumption and several outcomes. And current raw milk consumption means that the children in the study that at the time that the analysis was done were still consuming raw milk. And what you basically see in for asthma, all the studies grouped to the left of the odds ratio of 1, so there is consistent effect on a lower prevalence of asthma. Of current wheeze, it's a bit less clear, but of course, wheeze and asthma can either be infection or asthma related, and especially in these young children before the first year of life, you cannot diagnose asthma as such. So this may be a combined effect on asthma sensitivity and respiratory tract infection. Allergic conjunctivitis, which is an allergic reaction in the nose and also in the eye, and also sensitization atopy, in which you see a slightly more difference between the studies. But overall, all of these studies suggest that there is an association between the consumption of unprocessed raw milk early in life and a lower prevalence of respiratory tract disease. Okay, but if that is the case, then which components could play a role in this? And as an immunologist, when you start looking at milk, milk is really a very interesting fluid because on the one hand, of course, it's there to feed and support growth and development of very young children, but it is also there to help protect infants and cows, of course, also in early life. And when you then look at the composition of milk, it's very complicated, but a lot of the components that you find back in milk actually have a function linked to the immune system. When you look back at this graph that was also shown by the previous presenter, you can see that when you look at the components in breast milk, there are really a lot of components that you can link to immune function. It can be immunoglobulins, it can be tolerogenic components, it can be anti-inflammatory cytokines, etc. And the interesting thing is that even though a cow is a different species, we see basically most of these factors back in bovine milk. And of course, bovine milk is used as a starting point to make infant nutrition, which is of course one of the reasons to look into these. Now, I would like to go into a few different of these proteins and shortly review what is known of them. Now, lactoferrin is actually the protein that has been studied for a very, very long time because lactoferrin was identified as a protein that binds to iron, but it has a clear antimicrobial activity. So when you add it in an in vitro assay, you see inhibition of bacterial growth. We also know that lactoferrin has an immunomodulatory activity, so it modulates the response of the immune system, and in some cases we have seen very interesting effects on infection as well. And at the next level, what lactoferrin also does, it promotes the epithelial gut barrier layer. If you think about the intestinal tract, you have a lot of potential threats there in the form of bacteria, your own microbiota, and it's essential that your epithelial lining remains intact, because if it's intact, you are less prone to develop gastrointestinal infection. And lactoferrin can play a role in that. Now, in vivo studies, and that's in this case in human studies, of course, there have been a lot of studies done in laboratory animals in the past. But we have seen in human studies that on the one hand, in elderly women, lactoferrin can enhance dendritic cell function, and especially the plasmacytoid dendritic cells that are important in certain viral infections. Lactoferrin has also been shown to have an effect on respiratory tract infection in adults in a different study. And what's I think most relevant at this point, because lactoferrin is often used in infant nutrition, is that lactoferrin seems to be associated with a decrease in late onset sepsis and in necrotizing enterocolitis. And that's especially seen in preterm infants and children with a very low birth weight. In addition to that, it may also have an effect on the occurrence, but mostly on the severity of diarrhea. I picked out one data slide from really a seminal paper by Manzoni et al., which was published in the Journal of the American Medical Association. And what they studied is a range of outcomes in newborns in which they were fed a control formula supplemented with bovine lactoferrin, or a formula supplemented with bovine lactoferrin and probiotic LGP. And what they found when we zoom into the effect of the lactoferrin versus controls is that the presence of lactoferrin in the formula had a significant effect on sepsis, on fungal infection, on fungal sepsis, and on mortality. There was only an effect seen on mortality in relation to sepsis. But clearly, taken together, these data indicate that lactoferrin from a cow has an effect on the infant immune system in vivo, which is really noteworthy, I think. Then what you also have in milk is anti-inflammatory cytokines. And I would like to point out especially two of them, IL-10, which is present mostly in colostrum, which is really the early milk just after a cow gives birth, and TGF-beta, which is present at relatively high levels in bovine milk. And what is interesting is that the levels of TGF-beta in breast milk have been associated inversely with the prevalence of allergy in children. So maybe the TGF-beta can play a role in that respect. There have been a lot of studies in vitro study, in animal models, in epidemiology, but most often they have been done with human or murine TGF-beta. And so it's not fair one-on-one to compare this to the human situation, but what we do know is that the TGF-beta present in cow's milk is for 99% identical in its active form to the human form. And from these studies, we know that both TGF-beta and IL-10 are really key cytokines in regulating the immune system, so preventing excessive activation, as you will see in inflammatory diseases and in allergy. TGF-beta is also crucial in the formation of a good intestinal barrier function and in the induction of oral tolerance. Now, oral tolerance is very important because if you are born and you would have an immune system that reacts to anything that you ingest, you would have a continuous intestinal inflammation. And the induction of oral tolerance is a process that the immune system has developed to prevent that you actually react to everything you ingest. The oral tolerance induction is really quite important, which is probably the main reason why the TGF-beta is present in breast milk, but also in cow's milk. Now, if you study that in vitro, you can study the effect of bovine IL-10 here on the left by studying a dendritic cell, which is a white blood cell that when you activate it starts producing a lot of cytokines like IL-12. And when you do that and you titrate in IL-10, you can see that the activation of these cells is really inhibited quite strongly. So illustrating that it prevents excessive immune activation. At the right, you basically see the same thing for TGF-beta, but it's slightly different here. This is a study in which we develop these dendritic cells in vitro, either in the absence or in the presence of the TGF-beta, and then we looked at the resulting cell types if they were able to produce this IL-12 when you stimulate it with a microbial component. And what you see there, the MoDC, which means monocyte-derived dendritic cell, they actually are very good in responding to this bacterial stimulus like LPS or peptidoglycan, but the dendritic cells made in the presence of TGF-beta are not. So again, indicating that the components prevent excessive activation of the immune system and induction of inflammation. Another component which is really quite intriguing is immunoglobulins. Now, as was already explained in the first part of the webinar, it's really very important to have high levels of immunoglobulins in your blood and in your mucosal tissues like the intestines and the respiratory tract to protect you against infection. And in a newborn, immunoglobulins are transferred via the blood of the mother into the placenta and into the circulation of the infant. So at birth, an infant has high IgG levels, which drop in time by a factor 2 every 3 weeks after birth. And when they are breastfed, they are supplemented with IgA that's present in the breast milk. So that delivers IgA into the intestinal tract. Now, when you look at a cow, you have a slightly different situation. Cows mainly have IgG in colostrum, but also in mature milk. The ratio of IgG over IgA is about a factor 4, where in breast milk it's more 4 to 10 times more IgA than IgG. But an important question to ask yourself, of course, when you look at immunoglobulins from a cow and you're interested in human health, well, does it bind to the same pathogen? Yes, it does. And I will show you a little bit more on that later. Then another role of immunoglobulins is to bind to potential pathogens, but also promote the phagocytosis of these pathogens and clearance from the body. And for that, you need to know if the IgG binds to the receptor for IgG on human cells. And we have seen that in fact, bovine IgG can do that as well. We have also seen that when you look at respiratory syncytial virus, which is a common respiratory tract infection that can be quite serious in very young children, that bovine milk contains high levels of antibodies or globulin directed against this virus. And when you do an infection in vitro of human cells with human RSV, you can neutralize that infection with the bovine immunoglobulins. We also know from studies in the 90s when there were no combination treatments for HIV patients available, that HIV patients that often had recurrent diarrhea, that when they were treated with colostrum, which is very high in bovine IgG, that they really got better, and they went from like 7 stools a day to about 2 stools a day. So in that situation, high level of bovine immunoglobulin can protect against recurrent diarrhea in HIV patients. In infant studies, it's a different situation because until now, no studies have been done with purified IgG or immunoglobulins from just normal milk. Most of the studies that have been done in infants are with immunoglobulin preparations made from colostrum of cows that had been vaccinated against a specific pathogen, like, for instance, against rotavirus. And these studies typically are quite positive in preventing these infections. But of course, you have to note that in that case, you really have to go and vaccinate a cow with a specific pathogen, which is not really a normal approach for a, let's say, mainstream food product. So that's really for a clinical setting, not so much for a normal food setting. There have also been 2 studies in which a reduction in upper respiratory tract infections has been reported in which colostrum was used. Of course, we cannot conclude if that was the immunoglobulins in the colostrum, but it suggests that maybe also in that setting you might have impact of bovine immunoglobulin. Now, just to illustrate these things a little bit, what you can do is you can select a number of human pathogens. And as you can see in the graph, that's Escherichia coli, an enteropathogenic E. coli, Salmonella, or Streptococcus pneumoniae, which is linked to respiratory tract infection. And when you incubate these bacteria, which are the small dots you can see, with a control, which in this case was BSA, bovine serum albumin, you see that they are just lying there. So the BSA doesn't do anything to them. But when you incubate them with bovine immunoglobulin preparations, and that's mostly IgG1, as is denoted here, you see that you get aggregation of the bacteria or agglutination. And that is because an immunoglobulin has 2 binding sites and 1 constant region. So they can in fact cross-link different bacteria by binding to one bacterium with one end of the binding site and to another bacterium with the other binding site. And then you get these aggregates, which clearly show that it binds to these bacteria. Now, if the IgG binds to the bacteria, the next question is, can bacteria actually be taken up by phagocytes more efficiently because you have the IgG? And that's denoted here on the left. What you have on phagocytes like macrophages or neutrophils, monocytes, etc. These cell types, they have Fc receptors, which is a receptor that binds to the constant region of an immunoglobulin. So when you have a pathogen that is bound by a bovine antibody or immunoglobulin, that can bind to the receptor and then enhance uptake. Now that's the idea, and what we found, as you can see to the right, is when you take bacteria that are fluorescently labeled with FITC, and you incubate them not with any immunoglobulins, which is the zero, then you see that only a few percent of these bacteria are taken up. If you then expose them to IVIG, which basically is a human immunoglobulin preparation isolated from normal blood donation, when you titrate IgG into the system, you see the more IgG of humans you give, the higher the level of the bacteria that are phagocytosed is getting. So it means that, as you can see on the left, the IgG binds to the pathogen, and the IgG then is bound by the Fc receptor, and phagocytosis is induced. When you do the same thing with bovine IgG, you see exactly the same happening. You just need a little bit more when you compare the concentration of the bovine to the human IgG. And of course, that's not strange because a cow is not exposed to exactly the same pathogen that a human is, but still you do see the binding to the pathogens, but you also see the enhanced uptake and clearing in the presence of bovine IgG. Now, this was quite surprising when we started to look at in vitro infections with RSV, respiratory syncytial virus. We saw the following. I will take you through this step by step. If you first focus on the red line, that's a therapeutic monoclonal antibody, which is an antibody that is used in the clinic to treat children that are at high risk of RSV infection. And when you start giving that in the in vitro culture system, you see an S-shaped curve, but you end up at the level of 100% inhibition of infecting human cells with human RSV. Now, if you do the same thing with the orange dotted line, and you look at the human serum IgG, you see the same curve, but it's shifted to the right. Now, why does it shift to the right? This monoclonal antibody is a preparation of immunoglobulins that are all identical. They all have the same specificity, so they can all bind to the right epitope on RSV and prevent the infection. If you look at the human serum IgG, of course, only a fraction of the IgG is specific for this virus. So you need much more to get to this point of complete neutralization. But then, quite surprisingly, if you look at the bovine IgG, you get to the same level of inhibition by just adding, again, a little bit more. And if you look back at the phagocytosis slide and look at the right, you also there see that you need a bit more of the bovine, but the net effect can be the same, which you also see here. Now, the tricky and the interesting thing is that cows can also get RSV. And this means that in normal bovine milk, you get these immunoglobulins specific for RSV. And that's why you see these effects so clearly for the RSV. Now, when you try to add up what can a mixture of all these components in milk do, and of course this is a hypothetical representation, but let's say you ingest milk in which all these proteins are functionally active, then you can expect something at the level of the respiratory tract, because in the back of your throat, where there is immune tissue, you will be exposed to for example, to the antibodies that might play a role in the asthma effect. So you have a direct interaction really in the location where you expect or would like to see a clinical effect. And when you move into the GI tract, into the small intestine, there are a lot of components like the TGF-beta that can have an effect on the intestinal barrier function. And moving even further down, when you get to the large intestine, then it's more not so much about the proteins, but more about the indigestible oligosaccharides that can be present in milk, but also maybe the lactoferrin that can have an effect on the microbiota and can thus link to diarrhea. Now that's a great story. So when you look at raw milk, unprocessed milk, call it what you like, you have a number of these proteins that have an immune effect, and I should stress they have an immune effect across the species barrier. So in all of these cases, the proteins in cow's milk have an effect on cells of the human immune system, which also could be absent. But so far, in all these cases, we do see that. On top of that, in milk, there are a lot of other components, and some of them were already identified by Marlotte, like the vitamins, vitamin A, vitamin D are fat soluble. You have the milk fat globular membrane, which has been linked to decreased otitis in several studies. We have exosomes, which are membrane vesicles that contain messaging material in the form of microRNAs that can modulate gene expression. And then, of course, you have the minerals and additional vitamins that are in there. So it's really a very complex package of components. I think I didn't mention the sialyl lactose yet. That's one of the human milk oligosaccharides that is also present in bovine milk. And there are also components in milk fat that have an effect on intestinal barrier function as well via a receptor called AHR. Now that's a great story, but now comes practice. And practice is that raw milk and unprocessed milk can give you fantastic epidemiological results. But in the real situation, you need to come up with a safe product. And so you start with the raw milk, and then you have to go to a processing plant, and you end up, for example, with a milk powder or UHT milk. And the most important step in processing is heating to kill off bacteria and make sure it's safe. So the longer and the higher you heat, which you can see in the color-coded safety legislation box, the safer it gets. And maybe you even do an overshoot. So this means you adhere to legislation, you adhere to QA, QC, and to safety. You really have a safe product. But when you look at the functionality of the protein that I talked about, you get a different situation because a protein's function is linked to its structure. And heating basically unravels the three-dimensional structure of a protein and gives you a string of amino acids. And they are as nutritious as the starting protein, but the functional aspect is gone. So I think it will be a challenge to try to keep all these functional effects that you see when you start looking at the milk as it is produced by a cow to really a functional product in the end where it's still there. And there I think it's interesting to know that as FrieslandCampina, we basically control the chain from the grass to the glass. So we can talk to farmers, what food do they give to the cows. We can go there every day, collect the milk. And we know a lot on how to process the milk, to do it in a mild manner, to preserve all of these components. And we think by really following this process from start to the end, so from the grass to the glass, we may be able to offer a better, more nutritional, more natural nutrition of dairy. Now, I hope that what I've been going on about has convinced you a little bit, at least, that there are multiple studies that show an association between the consumption of raw unprocessed milk with a lower prevalence of allergy and respiratory tract infection. that at least part of the explanation of this, because it's in the raw milk and not when you heat it, has to do with heat sensitive components of the milk, which are the milk proteins, and that these milk proteins can have an effect on the human immune system, which is a relative thing to state. And then finally, I think towards the future, we are going to see more and more mild processing of milk with the aim to really try to keep everything intact that is in the milk to deliver its optimal functionality to the consumer. Okay, that's where I would like to stop, but as Marlotte did, we received a lot of questions by email. I can address a few questions shortly, and the rest of the questions will be answered to you at a later point via email. Now, one of the first questions we received was on my view on bovine colostrum in relation to immunity, and especially in relation to early life nutrition. Now, I think colostrum can be a really rich source of immunoglobulins because they are really high in IgG. It's about 100 to 1,000-fold higher in IgG than pure milk. The challenge is that not in all countries colostrum is allowed to be used in infant nutrition products. So that means that even though it's a great source, you cannot use it for that purpose. Now, as to the processing, which differences are there between the different types of dairy? And I'm looking at the clock, so in view of time, I will have to be quite short. I would like to focus on the unprocessed raw versus the pasteurized versus UHT milk. And then I think unprocessed and pasteurized, depending on the heat applied during the pasteurization, you will see something that is comparable when you look at the milk protein. In the UHT milk, you will not see the functionality of the milk protein. So in that respect, pasteurized may be the safe alternative of the three. Of course, given the current situation, a lot of questions were focused on the effect and the role of milk in relation to COVID-19. And I think, as Marlotte stated in the beginning, to get a well-functioning immune system and to maintain that, you need a well-balanced, healthy diet. And dairy can be a part of that healthy diet. But at the other hand, in relation to COVID-19, we cannot say anything about that because it simply hasn't been studied. It hasn't been around for long enough to study. So we cannot conclude anything in relation to COVID-19. We can only say something about milk being a part of a healthy diet. Now, as to the processing, I think I addressed it in the last part of my presentation. It doesn't have a lot of effect on nutrient content because if you inactivate a milk protein, it still consists of amino acids. So nutritionally, you don't see big changes, but you do see big changes as to the functionality of the heat sensitive component. That's not the minerals, that's not the vitamins that are heat sensitive, but that's typically the protein. So I think I have to sign off here. I would like to thank you all very much for your attention. And as stated in the last two slides, if there are additional questions, please ask them in the post-webinar survey, and we will try to answer them as well as possible. Thank you very much.